Good morning, everyone. We are continuing the second part of the chapter 9, Transport in Animals. So let's begin. So in the previous section, in the part 1 of this video, I have discussed about all the structure and parts of the human heart, the functioning of the heart, and I've also covered CHT and the causes and the reason behind and how we can reduce the chance of CHD. This video is the second part of that particular topic where I am going to cover all the blood vessels, arteries, veins, and the blood capillary. So let's begin. So in this section, we will talk about blood vessels. So majorly we have to study arteries, veins, and capillary as per the syllabus, Cambridge syllabus. So see, if you will see this diagram, this is the comparison of arteries and vein. So see, these arteries have a very narrow lumen and they have a thick layer of elastic fibers and smooth muscles because blood is moving with high pressure. So we need some kind of muscles, smooth muscles, which can slightly expand, and the elasticity will make it easy for the flow of the blood in high speed. And if you see this is the vein have the wider lumen and the smooth layer and the elastic fiber layer is very thin as compared to the arteries. Veins are one cell thick because they have to go in between the gaps of the cells to provide all the nutrients and oxygen. So this can't go because they are thick. Like how, um, take an example, like how the uh, water pipeline has been distributed in a city. So there's a main pipe, and then later on the pipelines distributed in different areas, and then from the areas it goes to the different home, right? All the houses with this thin I mean pipeline, not a thick one, right? Because the connection is going at home, so we need a thin pipeline. In the same way, arteries are thicker and they can't go in between the gaps of the cells, so that's why we have a blood capillary. Blood capillary helps uh, uh, to send all the nutrients and oxygen to the blood cells, and meanwhile they will collect all the waste substances from the cells. If we have to do comparison between arteries, veins, and capillary, so this is a very comparative um, slide where I have differentiated all the things in one place. It will be easy for you to memorize. So arteries carry blood at high pressure from the heart. We know it's coming from the heart, so obviously it will be high pressure. For veins, it's definitely low because it's going towards the heart. So they have to collect all the waste substances from the body to the heart. Capillary obviously low because they're going in between the gaps of the cells, so their pressure will be low. Arteries carry deoxygenated. Yes, there are some exceptions. So pulmonary artery is an exception here, which carry deoxygenated. Veins generally carry deoxygenated blood, except pulmonary vein. And capillary carries both oxygenated and deoxygenated blood. So there is no differentiation here. If you see the arrangement of the muscles and elastic fibers in the arteries, you will find that they are thicker and veins are thin walls, whereas the capillary are even thinner, they are one cell thick. Arteries have a narrow lumen so that they can maintain this flow of the blood. So area is low, pressure will be high. So there's the inverse relationship between pressure and the area. Whereas veins have larger lumen and capillaries are also nearby same, thin. The flow of the blood will be faster here. Here will be slow. And in capillary, it is again slow. There's one difference here, valves. Valves is present in the veins, absent in arteries, absent in blood capillary. But blood capillaries has a gap between the cells so that WBC squeeze out from the capillary and goes in between the gaps of the cells to destroy all the germs if it is present. Right? Structure and function of the blood vessels. So arteries have a thick muscular wall that contain elastic fiber. Why? To withstand the high pressure of the blood and maintain the blood pressure. So obviously we have to maintain the pressure because if anywhere the pressure is down, then blood is not going to reach to the cells. So it's very important task to make sure that it is flowing with high pressure. Obviously, they have a narrow lumen to maintain that pressure. Then veins, they have a larger lumen as blood pressure is low. Contain valve so that blood should not go back. Maintain the one-way flow of the blood. Then blood capillary have walls that are one cell thick so that substance can easily diffuse in and out of the cell. And they have a leaky walls means there is a gap between the cells so that the plasma or other tissues, blood tissues can come out. This is again I am showing you the arrangement of the arteries and veins. So question may be asked from this to identify arteries and veins. So even sometime in some of the questions you will find that the lumen size is same in arteries and lumen. How to identify artery and veins? See the thickness of the walls. So if you will find this is thicker, this is thinner. So obviously this will be artery, this will be vein. Arterioles and venules, what are these? As artery divide more, they get further away from the heart and they get narrower and narrower. So the narrow vessels that connects artery to the blood capillary, that area or that segment of the blood vessels is called arterioles. In the same way, veins also get narrow to the further away they are from the heart. So the narrow vessels that connects capillary to veins are again, we call them venules. This is a comparison slide. You can pause this video and you can check. Now, how the circulation going around the body. So if you can see here, this is what all names are important. First of all, we have to understand the names which is important here. So aorta, hepatic artery, this one, this is the hepatic artery going to the gut, going to the intestine. This is hepatic portal vein, connects intestine to the liver, and this is hepatic vein. This is renal artery, this is renal vein. This is pulmonary artery, this is pulmonary vein. Okay, so these are some points which is important. So blood carry away from the heart and towards the organ in arteries. These narrow to arterioles and then capillary and then pass to the organs. The capillary widen to venules and finally veins as they move away from the organ. So veins carry blood back towards the heart. Again. So this is how you have to remember. Mesenteric artery also called. So this name is important. I'm just telling the names which you have to remember. Pulmonary vein, aorta, hepatic artery, renal vein, hepatic portal vein, hepatic vein, vena cava, and pulmonary artery. So you can stop this video and you can make a flow of the blood vessels with the name. Important blood vessel here, heart towards the organ, vena cava and pulmonary vein, away from the organ, aorta and pulmonary artery. Lungs, is pulmonary artery and pulmonary vein away from the organ. Kidney towards the organ is renal artery, away from the organ renal vein. So you have to remember minimum these namings in the circulation uh, segment, this one. Then blood vessels and the liver. Again, sometimes they ask this section. So I'm just showing you this section part. This is the hepatic artery, which is carrying oxygenated blood from the heart to the liver. And hepatic veins is this one, the blue color, which carries this deoxygenated blood from liver back to the heart. So this blood vessel will go up vena cava and goes to the heart. Hepatic portal vein, this one, connects the gut to the liver. This transport deoxygenated blood from the gut to the liver. So this is also a kind of vein. Components of blood. Blood have uh, four components. So you can see red blood cells, white blood cells, and platelets, which is the majority in the blood component, and the 45%. Majority is plasma, and 45% is these blood cells. Without blood cells, plasma contain all the dissolved substances like glucose, amino acid, vitamins, enzyme, hormones, waste substances, urea, uric acid, and all that. Thrombin. So important is the structure you have to remember. So structure is unique in all three. So red blood cells look like biconcave disc containing no nucleus, and they contain hemoglobin. Whereas white blood cells, they are quite large. They have a big nucleus. Different types have different types of structure and functions. So we don't have to go in the detail of the different types. So we'll stick to the structure, large cells and large nucleus. Platelets is fragment of cells without nucleus. Plasma is a watery color liquid. Identifying red and white blood cells. So you can see here. So this is white blood cells because the cell is also big and the nucleus is also big, right? This is again white blood cells. These red colors are normal red blood cells, and these are platelet. Easy to identify. If you have to identify between lymphocytes and phagocytes, which is the two type of WBC, so this is phagocytes and this is lymphocytes. So lymphocyte is a big cell and one nucleus, whereas phagocytes have big cells and nucleated cells. What is phagocytes? Phagocytes actually they are type of WBC. They perform phagocytosis, meaning is engulfing. First they engulf the bacteria, and phagocytes secretes digestive enzymes which digest the pathogen. And they are highly sensitive to the cell membrane that can detect chemical produced by the pathogenic cells. So once pathogen will do something in the cell, these phagocytes will get triggered and they will start releasing digestive enzyme. They can easily recognize under the microscope by the multinucleated lobe, and they have a granule cytoplasm. So you can see here in this diagram, multinucleated and granules are also there. This is how the phagocyte process takes place. This is the phagocyte, this is the cell, engulfed by phagocytosis, and then enzymes will destroy the bacteria. Lymphocytes, they have only one functions. They produce antibodies. They have a large nucleus and they are non-granular cytoplasm. There is no granules. What is the function of the blood? Plasma has only one function, that is transportation of the blood cells, nutrients, urea, minerals, hormones, antibodies, and carbon dioxide and blood cells. What red blood cells do? They transport oxygen and the respiratory gases. What white blood cells do? They perform defense mechanism, prevent body from any infection. Platelet involving clotting of the blood. How blood clotting takes place? So the platelets are actually fragment of cells, and uh, this blood clotting process continued until unless the flow of the blood has been stopped. The platelet actually releases a chemical that causes soluble fibrinogen protein, and they convert it into the insoluble form, which is called fibrin. So fibrinogen is converted into fibrin first. Fibrin is insoluble form and fibrinogen is a soluble form. Now this insoluble fibrin will make a mesh-like structure, and they trap all the red blood cells, and then later on they will form a clot. In this way, it performs two things. First, stopping all the blood cells to come out, and second, also stops entry of microorganism, and it will prevent infection. So conversion of fibrinogen is not easy task. Easy task means it's not controlled by, it is controlled by many signals actually. So platelets are fragments of cells that are involved in blood clotting. So, so first of all, when the skin is broken, so platelet will reach to that particular point where the bleeding is going on, and a series of reaction will begin. So a type of cascade of reaction you can say, where the fibrin is converted into fibrin and it will create a mesh. And then later on, after a few hours, you will find that clot is dried up and they form a scab to protect the that wound from the bacteria. So this is how this is the cutting. If it is not being clot, then the blood cell will keep on going. This is fibrinogen, the red colored things. They are coming and they're trapping the red blood cells like this. So in phase step, blood clotting process stops. This is all about chapter 9. So we have completed the transport in animals. Thank you everyone. We'll see you with the next video.